So my name is Laura Saba. I'm co-director of the NIDA Center of Excellence in Omics, Systems Genetics, and the Dictome, along with Rob Williams, who'll be one of our two presenters today. So this webinar series, which we've entitled Quantitative Genetics Tools for Mapping Quantitative Trait to Mechanisms, Therapeutics, and Interventions. So we are officially in webinar 21. The other 20 webinars, including the one from 2 weeks ago by Carl Broman on organizing data in spreadsheets, are all up on our YouTube channel and can be accessed through our website, which is called OPAR. Maybe I can get Rob or Xusheng to write our website in the chat for me. A few housekeeping things before I hand it over to Rob to do introductions. First of all, you're automatically muted when you come in. We appreciate it just because of the number of people on the call. If you stay muted during the presentation and write your questions in the chat, Xusheng's going to be watching and monitoring that chat for us. And so when he sees questions that warrant clarification right at the moment, he'll go ahead and speak up. If there are more discussion related questions, we have designed this webinar to be in general an hour of presentations, then followed by 30 minutes of discussion for those that can stay online. It usually takes us a little less than a week to get the video up on the website and all the appropriate links there after it is. So if you have to leave early or if you want to share it with a colleague, we will put this recording up on YouTube as well. I think that takes care of the housekeeping things. I'll go ahead and hand it over to Rob to introduce Xusheng.
Yeah, it's a great pleasure to have Xusheng join us to give a presentation. He's been a regular at our OPAR webinars, and great, Xusheng, thanks for agreeing to do this. Xusheng was also a recipient of a pilot grant from the OPAR P30, and it went so well that he's taken on a larger role in our pending renewal. But I just thought of an efficient way to do an introduction and not make too many boo-boos is actually to show you his bio sketch. And he started out in Xusheng on the east coast of China, just a little bit south of, well, probably a couple hours south of Shanghai. It was sort of a unique university that had a joint PhD with the University of California, Davis, where I got my PhD. And so he has always had, since I've met him, he's always had excellent English, which is wonderful. He's a really strong bioinformaticist. Xusheng is famous for its bioinformatics and genetics, statistical genetics programs. So he came, when he came to UT in 2008, he really was more than a postdoc. He was actually responsible almost single-handedly, poor Xusheng, for all of the genomics and bioinformatics we did on campus. So he was sort of a one-man band for many of us. And he stayed at UT, came out with a really high impact set of papers with Dr. Lulu and myself and others, Megan, I don't know how many others, but really very productive stay here, and then moved down the street to St. Jude, where he worked for the first time very intensely in proteomics with Junming Peng, who is a famous proteomics expert who was at Emory, now at St. Jude. And I think Junming even, if I'm correct, Xusheng did a lot of work with Geige at Harvard at some point. So Xusheng really pushed the envelope on proteomic workflows and software development. And this whole JUMP series that you see here, JUMP for peptide identification, JUMP G for proteomics, that's all work that he's done. So in addition to doing the analysis, he also builds the tool sets that are critical for proteomics. So we couldn't have had a better expert to give us this talk. The great thing from my point of view is he's worked with the hybrid rat diversity panel, and he'll be telling you a little bit about that data set. towards the end of his presentation. Then I'll be giving you a short tour of the data that Xusheng and team generated, and then I'll hand it back to Xusheng. So thanks very much, Xusheng, for joining us.
Thank you very much, Love, for your nice introduction. So this is my screen. Can you see my screen?
Yes, but it's not yet in presenter mode.
Okay.
There you go. Perfect.
Yeah. So today, I think Love and I are going to present proteomics and the protein-QTL analysis. We will first give you a general introduction to proteomics technology and data analysis. And then we will present a new pipeline that we developed for correcting sample mix-up in proteome data. And finally, we will discuss red brain proteome and the protein-QTL. So first of all, what's actually is Massbackbased proteomics? And for a typical Massbackbased proteomics workflow, and protein are first extracted from the biological samples. And in some experiment, you want to enrich, organelles or cell type, but it is optional. And protein are first extracted from the biological sample, then it digests into the peptide with enzyme such as trypsin, and the mixed peptide then separated by HPLC, and the peptide are ionized and inject to the high resolution mass spec. And what you will get is two types of information. One is MS1 spectrum, and then MS2 spectrum. So MS1 spectrum contain all the information about the mass and intensity of the peptide, and MS2 spectrum contain the mass and intensity of peptide fragments. And for the last step of the proteomics is the data analysis. So I will introduce the data analysis later on. And so there are several approaches for quantifying peptide and proteins. And so this approach can be divided into absolute and relative quantification. In absolute quantification, we want to determine the copy number or concentration of the protein within a sample, whereas in relative quantification, a ratio or relative change of protein concentration between sample is measured and calculated. Both absolutely and relative quantification can be done with either label-free approach or labeling approach. So for label-free approach, a big challenge for label-free approach is batch effect and data normalization. And so, as you can see, each sample is analyzed by LCMS independently from the sample preparation to data acquisition. So it suffers a big variation between sample to sample. And so for labeling approach, there are chemical labeling approach and metabolic labeling approach. So metabolic labeling, typically the method labels arginine or lysine with a stable isotopic, and it's labeled on the protein. So this approach is the most accurate. But the downside for this approach is they cannot multiplex many samples. For example, the SILAC approach only measured up to 3 samples for one experiment. And for the isobaric labeling, And it can be labeled at protein level or labeled at peptide level. But the good thing for this approach is you can measure 18, right now up to 18 samples at the same time. And this is a big advantage compared to the other approach. But the downside for this approach is the ratio suppression because of co-eluting peptides. And so for some of the spiking peptide, you can spiking the peptide into the sample, you can measure the absolute quantification. And so the most widely used chemical labeling approach is a TMT-based approach. And as I said, we can label 18 samples right now, but here I just take a 10 sample, 10 TMT, a 10-plex TMT experiment as an example. And for example, you have a 10 biological sample, and it digests into peptide, then you can label it with a peptide with a TMT reagent. And after that, you can mix all these peptides and across all the samples, and then you separate the sample the peptide into different fractions using HPLC. And then you can take one fractions and inject the mass spec and get the data. And so for data analysis, and I think there are 4 major steps involved in proteomics data analysis, including peptide identification, peptide filtering, and protein quantification, as well as pathway analysis. As Rob mentioned, we actually developed a nice comprehensive pipeline called the JUMP suite, and which include peptide identification. And for the JUMP, we actually able to identify more 20% more ID than to widely used approach, like a sequence and a mass card. And to further increase the identification rate, and we developed this TMT spectrum library, which also allow us to increase 20% ID rate. And we also developed, JUMP-G, proteogenomics pipeline, which is able to integrate genomics transcription data and proteome data to identify novel peptide or variant peptide. And recently, we also published a tool called the JUMP-N for co-expression analysis for proteome data. For database search, we first download protein sequence from public resource such as UniProt and NCBI or UCSC genome browser. And then we can do in silico digestion and make all these peptides. And the search will typically calculate this theoretical mass for all this in silico peptides. And then for each spectrum we get from experiment, the search engine will calculate a matching score and basically match this measured mass against all the theoretical mass in the database. And we will identify the best matching for a spectrum. But the best matching may not be the true positive. And so we typically control the FDR using the targeted decoy strategy. In the targeted decoy strategies, spectrum are searched against, not only against this target database, but also to decoy database. And at the end, we will calculate the distribution of targets peptide and the distribution of decoy peptide. And Then we define the FDR by a ratio of force target, which is a decoy, the number of the decoy, divided by target. Sorry, this should be target. And to generate the decoy, we basically use the target sequence to reverse the sequence into the force protein sequence. Then we do the digestion. And so this is a target decoy approach we use for FDR control. So as I discussed, and what we measured is a PSM level, which means peptide and spectrum matching level. However, we are interested in the expression level at the protein level. So we need to summarize the protein, the expression level from PSM level to protein level. What do we typically, we first summarize the expression level at the peptide level first, and then we summarize the expression level at the protein level. So at the end, we typically have 3 tables. One is PSM expression level, and the peptide level, and the protein level. So with this developed proteomic technology, we recently generated human brain podium data. For this data, we include 210 normal control samples and 49 schizophrenia samples and 29 bipolar samples. After we get this sample, we realize this sample has a severe mix-up issues. And our crossvalidate actually make this correction for other sequencing data, such as riboseq and atac-seq. And as I said, so proteomic data cannot provide those variant data or genotype. And but what we get is all this spectrum. And then how can we get the genotype from this spectrum? So with that, we recently developed this SMAP pipeline for sample correction. And SMAP actually contains 3 components. The first component is variant peptide identification using proteome genomics approach. And the second step is inferring the genotype for each samples. And the last step, we just do the scoring and the verifying sample identities. And to illustrate the idea of the SMAP, let me just give you this example. This is a typical spectrum from the TMT-based proteomes. And so this is MS2 spectrum. On the left side, this peak calls a reporter ion, and which is used to quantify the expression level of the peptide across the samples, in the samples. And this peak on the right side is used for peptide identification. If you zoom in this one, right, zoom in this reporter ion, as you can see, the sample 3 has a high expression, but has a low expression in all the other samples. And for this for the peptide identification, we curate this database by including all this variant peptide as well as all this canonical protein sequence. And by searching this spectrum against this database, we can identify some variant peptide like this. And with this variant peptide, we can get back to the allele information. So for example, this reference allele is A and the alternative allele is G. So then based on this expression data, we can say this sample 3 has this alternative allele, and all the other sample has a reference allele A. because this peptide is a variant peptide. And if this is still not clear to you, I probably let's look at a more close about this example. So for example, you have a SNP on chromosome 1 with this reference allele A and the mutant allele G. So the reference allele will generate this peptide, reference peptide, and this mutant allele will generate a mutant peptide. And if you have a sample, 10 samples, so the sample 3 has this mutant allele, which will be observed this variant peptide, because all the other example has this as a reference allele. We will not observe this reference, this mutant peptide. So if you look at the proteome data, and you will see a highly expression data in the sample 3, but you have very low expression data for all the other samples. So ideally, you should not be able to observe any of these signals. But proteomics, Massback basically generate a lot of noise. And so this is basically noise level, and which also makes this our analysis a little bit more complicated. And so, but overall, you can tell the sample 3 has a mutant allele, but the other sample has a reference allele. So again, so the peptide, when you measure the peptide, you typically will generate very significant different magnitude of the abundance for different peptides. But in order to characterize this reference allele and mutant allele, we first standardize the expression level into a range of from 0 to 1, with the minimal signal as 0 and the largest signal as 1. And with this, we classify this example into 3 categories, and using this quantile approach. And so the first one for this first quantile, and which is a reference allele, and the above quantile is a mutant allele, and in the middle intern quantile, which is a heterozygous allele. And so with that, we can infer all the SNPs for each examples. And then we compare these examples against all the genotype, the example in the genotype data. And we calculated 2 scores. One is a matching score, or we call the concordance score. And another one is a delta concordance score. And the matching score means, the ratio of ratio of the matched genotypes. And the delta score, which is measure the first hit minus the second hit divided by the first hit, and which is a kind of how specificity of the matching, and which is actually also give us very good indicator to separate it from the true hits from false hits. And here is the result from one batch. And as you can see, there's one example is mismatched because our SMAP give a higher matching score and a higher delta matching score. But the example ID was mislabeled. But for all the other examples are correct and match to original samples. And you can also say the internal standard, which is a mixed example, it just gives you lower matching score and lower delta score, because this sample is the genotype of this example is a mixture. And then we also applied all this, our SNAP program to all the 288 examples, and we identified 54 mislabeled examples in our data. And to 12 example, we cannot correct it through our approach. And as also you can see, those internal standard also show very, very low matching score and the delta score. So then we performed a cross-validation about our correction with other omics correction. As I told you, we make correction for the ATAC-seq and the riboseq. And among these 54 correct example, 34 example actually was correct across 3 platforms, including Massback and riboseq and ATAC-seq. And 5 examples were correct between by both Massback and riboseq, and 9 Massback and ATAC-seq, and also have 6, which is the Massback unique platform specific correction. And if you look at all this, we're looking into the more close about this swapping and how this mislabeling occurred. And the vast majority are actually pairwise swaps. And we also observe one example that is a loop swap. And so for our data, we also do some simulation to see how many SNP is enough for SMAP to correct examples. And we basically simulate data by shuffling the SNP in example. And as you can see, if we shuffle 60% of the genotype, we're still able to identify the right examples. If you have more than 60 shuffled SNP, you were not able to identify the right examples. And we also, cross-validate with other tools. And recently, tools published by Bing Zhang group, using expression, expression level, when they this tool called the Cosmo, they first select the features, and then those features are highly correlated protein and genes across all the examples, and then they use these features to see whether this is a match examples. And after we also apply this Cosmo to our data, and about 94% of the examples are consistent correction made by SMAP and Cosmo. And but we found that SMAP has a full correction, but Cosmo make misadjustment. And also Cosmo made 2 right adjustment, but SMAP didn't adjust it. And they have 2 examples, both were assigned different ID by Cosmo and SMAP. And about 10 examples, both approach cannot give right examples. And so at the final step, I will briefly discuss this red-brain podium data. And in the early this year, we profiled the red-brain podium from this HXB and VXH gene. And so we generated this proteome data from 21 strains, including 2 parental strains and 19 RS genes. And so using 3 batches of the 16 plex TMT, and at the end we generated 8.3 million spectrum. After database search, we identified 99,000 peptides which correspond to 9,000 proteins. And among this 9,000 protein, 8,000 protein were quantified across all the samples. And but the vast majority of this protein actually were quantified across all the samples. And we also, when we compare the protein expression with the RNA-seq, and we found the 70% of this highly abundant gene also detected by Massback. And because we include the two pentose gene, and so we can detect the DEG, DE proteins between two pentose gene. And so we found that around 177 proteins at 1% FDR and 4-fold change. Here is an example. For example, this ATF3 shows a 4-fold change between SHR and BNLX. And then with this data, we perform a pQTL analysis. And so at the end, we identified 222 cis pQTL. And we also applied the same procedure to the eqtl. We download the transcript data from Phenogene database, and then we identify 2,600 cis eqtl, and we summarize this pQTL and eqtl in this circle plots. And as you can see, some of the region, for example, here, they are shows a consistent pQTL and the eqtl. And so next step, we actually assess how many eqtl and pQTL are co-localized. And we use one program called the co-loc program. So the idea of this co-loc program is we can summarize this idea using this diagram. So if we use one as a significant QTL and 0 as a non-significant QTL, so then we will have either no association for either pQTL and eqtl, or we have association in the pQTL but not in the eqtl, or we have association in eqtl, not a pQTL. The first case is you can have both significant QTL, but they have a different SNP control the QTL. And the final case is they have a same association, but they also have the same SNP control the expression. And with this assumption, and they calculate the probability. At the end, they calculate a posterior probability for this PP4, this assumption. If this assumption is a largest probability, and then they think this is a co-localized QTL. So as I said, we detected 222 cis pQTL and 2,600 cis eqtl, and then we look at how many protein and the gene pairs are within the 1 megabase distance. And then we identify 101 pairs and analyze by co-localization program, and we identify 61 co-localized eqtl and pQTL. And here is just to give you one nice example, and this is shows this eqtl here and the pQTL here. Both are controlled by the same SNP. And here, this is one of the nice trans-QTL from this proteome data. I will pass this one to Rob, and Rob will tell this nice story about our trans-QTL.
Thank you so much, Xusheng. That was great. And I just in the chat, what I thought was great about what you presented is it was a really good summary of the alternative methods for doing the proteomics. But the beauty for me was the recognition of the importance of error control at all stages of these very complicated protocols. So proteomics has been one of those poster childs of almost there for the last 20 years. It's finally there. So proteomics is now a quite mature technology and the price has come down. And I think Xusheng will have some time to talk about that in the discussion section. But I'm delighted to see the technology come so far so fast. So I'm going to share my screen and we're going to do a dive, not too deep a dive into Xusheng's data set. So I'm just opening up a browser and anybody that wants to do this can also do this. I'm going to genenetwork.org, which is a web service that's integrated in the P30, but it's a web service that's also funded independently to Shanak Sen and Piotr Prince. It's actually, I may have said this before, sorry, it's getting probably boring to hear it from me. It's actually the oldest web service in biomedical research. So it started in 1994 as the portable dictionary of the mouse genome, and now it has a lot of human data. So GTEx, for example, is all of there. In the human data, you will look long and hard and you will not find any proteomics data yet. So if anybody knows of genetic data, genetic proteomic data, please let me and Xusheng and Laura and Shanak know because we'll put it in there. So I'm going to switch over to RAT, where we do have the impressive data that Xusheng and team have already generated. It is in the hybrid rat diversity panel, which includes CHXB, BXH. It's not a trait or cofactor. So trait or cofactor would be something like body weight or a classic phenotype like response to cocaine or morphine. It instead will be down here in the molecular traits, AKA endophenotypes. And what you just heard about is one of two data sets that Xusheng has put in here under brain proteome. I don't think time will permit, but I'm going to show you, I'm going to include a little document for you so you could look at the brain metabolome. This is the data that Laura and team have generated over the last 5+ years with Boris Tabakov and colleagues, Paula Hoffman. It's a really beautiful RNA for the whole brain. Some of these are older data sets that our group here generated for mRNA. A lot of these are STAR consortium data sets, but this is our data set of the day. I'm going to make it my default. Before I dive into it, I just want to show you that there are actually two levels here. So Xusheng mentioned both the peptide level and the protein level. The transform is very important. In Xusheng's QC and genotyping call, he used a log scale. Doesn't really matter which log scale you use. Log 2 is somewhat easier to interpret. A 1 unit difference is a 2-fold difference in expression. We're not quite finished, so I hope you'll have a late 2021 data set or maybe a 2022 data set, which will be slightly larger. I am going to now share a document with you in the chat. So make sure you've got your chat window open, and I'm going to send you a not very long Word document. I hope most of you can get that. I still consider, unfortunately, still consider Word to be our lingua franca. So have a look at that if you want to basically do what I'm about to do. And what I am about to do is basically go through this document. Let me open it up just to give you a feel for it. So actually, I think I might have given you the old version. Let me get the new version. I'll put that. This is the new version I actually want to give you. So let me update that. You can, if you downloaded that one, throw it away. I'm giving you a slightly beefier version that doesn't have as many typos. So take that second one. And let me open it. So here is what that'll look like. It probably is something, the good thing about having this on a Friday is now you can spend all of your Saturday and Sunday going through this and making sure you can do some cool things with proteomic data. And if you're interested in the metabolomic data, there's a little bit about that. So I will keep that in the background in case Gene Network does something persnickety while I'm talking. But let me show you how to use these data sets. Hybrid rat diversity panel, proteome, choose your level. So if you're really interested in peptide level, as Xusheng mentioned, there are about 100,000 peptides in there and about 8,500 protein assays here. I and many of you that are interested in substance use disorders will be interested in a large number of proteins that are, for better or worse, for today, are membrane fractions, or membrane-bound proteins. If you look for DRD1 or DRD2, expect to have a little bit of a heartbreak. If you're looking for a GABA receptor, expect a bit of heartbreak. But here's a way to overcome some of that problem. There's some advanced commands down here, and I'm going to use this advanced command. I'm just copying it. I'm going to put it in the get any. I'm not interested in mitochondria today. I'm interested in dopamine. 1001, 1002. If I'm not competing with too many of you for using the service, this should take about 10 seconds. It's looking through every one of the proteins, comparing it to RIF. RIF stands for reference into function, and asking, do any of the reference into function entries in NCBI for gene X, Y, and Z have the word dopamine in them? And while it's working on that, I will show you what the result will be, just in the interest of efficiency. So this is basically what we did. In this case, I actually did a slightly more sophisticated search. I did a search that combines two terms, the one that I just introduced you to, RIF equals dopamine, and LRS equals 15 to 999. That means that the LOD score, or the minus log P, or the likelihood ratio statistic has to be between 15 and 999. 999 is a very high number and will not be a problem. Let me try to literally do that and see if it's still cached somewhere. So I'm just going to do what you see there in the document. RS equals between the range of 15 and 999. And we'll do that more complicated combined search. And I will let that run in the background with a little more patience than I did. So what this should do, if I'm, again, not competing with too many of you, and in case I am competing with too many of you, I'm going to do something a little bit tricky. I'm going to go to another service. And go here. I'm going to make this my fault. So this is a staging system we use. Okay. So I just am not quite patient enough. So this now found 115 records in that particular database associated with the gene RIF containing dopamine and a pretty good linkage statistic where 15 is sort of low, but reasonable. And these are the names of the genes or proteins, actually. So these are actually protein symbols, or they should be all capitalized. The location of the gene, cognate gene of that protein, the mean expression level in this log2 space that Xusheng introduced you to, the location of the peak log score or minus log p, and where that peak is located, and the effect size. The little I here will tell you what the heck is an effect size. I'm going to finesse that for now. I'm going to do something pretty crude, which is called phishing. I call it trawling. So we're going to trawl for all of the good genes that are linked to dopamine and have very high statistics. So these are log scores above 6. And what's interesting is those first 4 all map on chromosome 1 at roughly 44 megabases. What is going on? So Xusheng showed you that he can systematically map all cis eqtls and trans eqtls. So the first question is, are any of these proteins cis eqtls? So the endothelin B receptor, the gene is located on chromosome 15 at 88 megabases. The peak is on chromosome 1. So that is actually a trans eqtl or trans pQTL. P for protein, but E works equally well in my book because it's an expression of a protein, and the E does not mean anything other than expression. And protein, metabolites, peptides all have expression. Chromosome, so the second one here, Synaptotagmin 7, chromosome 1 at 226, also trans. So this is the trans band that Xusheng showed you, and let's play with it a little bit. I'm going to pick the second gene on this list, and we're going to treat it like a dog would treat a good tasting bone. So here it is, Synaptotagmin 7. We want to know a little bit about Synaptotagmin 7. We could go to another link over here, the rat genome database, Phenogen. I'm going to go right to the gene wiki and see what it says. So it's a presynaptic protein, high in Purkinje cells, excellent candidate for brain weight in certain data sets. Where did the word dopamine come in? There. So I just did a search for dopamine in here. These are all the reference and function entries, gene riffs. So somatodendritic dopamine release requires synaptotagmin 4 and 7, voltage-gated calcium channels. And if you wanted to read the paper, you could do that. Is that the only appearance of the word dopamine? Yes. It's the only appearance of the word dopamine. But let's take it anyway. So let's look at the data. So now I'm just giving you a little overview of what we call findable, accessible, interoperable, replicable, robust, what have you. So fair data. 21 strains that Xusheng told you about in the second half of his talk. Histogram of the distribution. You'll notice the counts are still low, right? We only have 21 cases here. If this were based on 200 cases, we would talk about a bimodal distribution. We can't do that here, so this is just a sampling artifact, probably of the way the histogram was constructed. But we can ask, what is the skewness? What is the kurtosis? Is it sharper than expected or flatter than expected? Is it skewed in one direction or another? It is a little bit flatter than expected for normal distribution. So that's kind of an interesting finding. Let's look at the bar charts for the 21 strains. So there they are on, this isn't even going down to 0. So this is a fairly small difference. Notice that we're truncated here at a value of 8. If you put this, if you click on this edit in Chart Studio, you can change this figure any way you see fit. It'll open up the data in Chart Studio, totally free of charge, lets you do some cool things. You can see that 2 of the strains are pretty noisy, maybe 3 strains. But in general, that is not terrible data. You can look at the probability plot, and you'll see that little jump there that we noticed in the histogram. And let's look at it as a violin plot. Again, a little bit broad. So let's map it. So we know that this maps well because that's pretty much why we picked it. It does not have any blatant outliers we have to fix, so I'm not going to do anything to the distribution, although I could. So there are all kinds of options here for fiddling with the data. And you should fiddle with the data. Do not be afraid to fiddle with the data. It's really important in some cases. If you don't fiddle with the data, there's a very real risk that your mapping will go astray. So please be bold and do not consider the data sacrosanct. It's not sacrosanct. We're going to go to mapping tools, and I'm going to map this with a linear mixed model that is quite powerful, but you could map this with eqtl1, eqtl2, Haley knot, etc. But I'm going to map it with this. I'm going to use the super new genotypes that Hao Chen and Hakan Gunterin generated from sequence data. It's experimental, but it's actually quite a bit better than our not so new. So let's map it. It gives you good metadata about what it's doing. It does it so fast that that it didn't even update the second, probably because it was cached. And what you see here is our QTL peak on chromosome 1. It's not quite the same value we had in the over here. Remember, here we had a value of 6.8. This was computed using a different code. We just computed it with GEMMA, which is a more accurate, better way to map the traits. So the value here isn't exactly the same, but the location is the same. This is the location. This purple triangle right here shows you the location of Synaptotagmin 7. This is the location, the peak way over here. They're not linked at all. They're both on chromosome 1, but they're not linked. So let's zoom in to chromosome 1. And I have about 2 more minutes that I can show you how to do this. But I will show you that in this particular case, we have not a good candidate, but an absolutely fantastic candidate. These are all the genes on rat chromosome 1. Here's our QTL peak. It's not tiny, but it's not huge. Let's zoom on it. I'm going to click right here on this purple or pink area to zoom on it. Now we're getting there. So this is our interval. There are about 100 genes there. So we're going from 35 megabases to about 43 megabases. Did I say 32 or 35? I said 35. Okay, so that's, whoops, didn't mean to do that. Let me just do this again, sorry. Okay, so I want to go to 35 here. And I want to start scanning for candidate genes. ICE1, I'm going to ignore all the unknown, unloved genes that don't have good names. You can see the human equivalent over here on chromosome 5. I'm going to skip over some genes. That's obviously interesting. A big gene with lots of variants, probably. Aren't. Estrogen receptor. Okay, that's kind of cool. Another synaptic gene, VIP. Cool. This, yes, interesting. Okay. End of story. So, I mean, it's not literally the end of story, but there's the mu-opiate receptor. And obviously, I'm a huge leap of faith, but the mu-opiate receptor is located right here. The mu-opiate receptor is, as you'd expect, a ginormous gene. It's 250 kilobases. And it is something that if you talk to Hao Chen or others, they can actually give you a list of all of the sequence variants in that region. So we cannot pretend that we've cloned the mu-opiate receptor as the modulator of these transcripts, but I can definitely tell you that it's a hypothesis, and it's a well-structured hypothesis with a causal basis. And the question is, well, is it really mu-opiate receptor? If we did a CRISPR-Cas9 and converted a B strain to an H strain or an H to a B, would we see modulation of these transcripts? That's about a 2-year project, but there might be some clever bioinformatic ways to avoid that. So I unfortunately have to leave now. So I'm going to pass it back to you, Xusheng. And thank you, everybody.
Thank you, Rob. Are there any questions from the audience? I don't see anything in the chat yet.
Xusheng, a great talk. I also have to leave right away, but I'm wondering if I think you have a fantastic pipeline for detecting SNPs. Is it possible to detect splicing variants as well?
Yes, yes.
Cool.
As long as you curate splicing variants in the database, we were able to detect whether this variants, splicing variants in the proteome data.
Great. Okay. Got to leave.
Any other questions? Any question about the sample preparation, or price or cost, or any?
I've got a question, Xusheng, to kind of get us started here. Can you talk a little bit about the batch effect? So when you did this, you did several large groups. Where were your biggest sources of variation? How would you best design the study to do that many samples?
Right. So batch effect can be contributed by a lot of sources. So like if we're doing brain, right, human brain for the data, for the data we generate. And there's a bigger batch effect for brain bank. And they also have a quality, sample quality. And for example, some of the quality has a broader contamination, and they clearly show the batch effect. So normally we can correct this batch effect by internal standard. Oh, we're using the PCA or a method to correct. And for these 288 examples, we even corrected by the internal standard, and we still cannot fully remove all this confounding effect. Then we use one of the tool called the PEER, developed by the Richard Dubin group. And we try to remove all the hidden confounding effect. After that, we get a pretty good mapping result.
Any other questions from the group? I've still got a couple more, but I'll leave it open to.
So tell us about the cost.
Oh, yeah. So it's dependent, right? If you want to send your sample to a company or some service core, it's roughly more than $500 per sample. But if you're doing, by our lab, if you have a technician or you have a person doing all this experiment, maybe it costs $300 per sample, $200 to $300 per sample, exclude without considering this personnel cost. And it also depends how deep you are going to do. Sometimes if you want to generate 3,000 or 4,000 proteins, and which will cost much less than you generate 10,000 proteins.
So, kind of another question is, we had a talk about metabolomics earlier in this webinar series. Metabolomics and proteomics are both using mass spec. How are these, what are the sort of similarities and differences in the way you do proteomics versus metabolomics or lipidomics?
Oh, of course, first of all, the sample preparation is different, right? One is extract the protein, one is that you have to extract the metabolites. And I would say proteomics technology is more mature compared the metabolomics. And right now, for the metabolomics, we cannot do, labeling, especially for the human sample or any other sample. So most of the time, we only do label-free approach. And in the metabolomic field, most of people still doing target approach. And when we say target, and you have a standard components first, and then you detect this your sample match to the standard to see whether this compounds in your sample also match to the standard based upon the mass and the retention time. So I would say proteomics technology is much more mature than metabolomics data. And normally you only get maximally 500 ID for the metabolomics. And most of people, you only get 200 ID.
So in the proteomics, as I understand, there's also this deconvolution step, right? You're breaking it into peptides, then reconstructing it. Mm-hmm. Are they doing the same thing in metabolomics?
Metabolomics don't have this issue. Metabolomics just detects the compound. And proteome, because we measure the peptide, we have to break the peptide into a protein into peptide. So we have to bring back to the protein level. And also there are a lot of the sharing. And but for metabolomics also has a isomer. They share the same mass but different structure of the compound, which is still mass spec cannot resolve this isomer. If you want to resolve this isomer, it's better to using NMR or some other technology.
Mm-hmm.
So, piggybacking on this idea of going from peptides to proteins, how do you handle it? So you showed that those peptides that have the variant in it, and you quantitate the different variant, you get different values. How do you handle that when you summarize into proteins?
Right. So as I said, we first summarize at the peptide level first, right? Because one peptide can be measured multiple times. And then, in order to accurately, quantify this protein, we typically only select this most reliable quantification for the peptide. For example, the top 3 measurement for each peptide, and then we summarize all this, get an average or media value for peptide. Then we get another media value or average value for the protein. But again, there's some issue in the proteome. This is kind of elephant in the room. So there's a shared peptide. For example, this peptide actually contributed by gene family, right? It's not contributed by contributed by this isoform. And so you can use the unique peptide, for example, the unique peptide from that isoform. And you can say, oh, this expression is represent this. But right now, people just roughly summarize all the peptide together to represent the protein. So right now, we still call the protein group, not the protein, really protein isoform.
Xusheng, I missed your entry point for the mislabeled samples there. Did you have genotype data and the proteome data from these people, or?
Right.
I missed the point about mislabeling. Say if you did, then I understand.
Yeah, for this data set, we generated all the whole genome sequencing, RNA sequencing, ATAC-seq, riboseq, and proteome data. So we also observed mislabeling in different data set, and we do the independent correction for the ATAC-seq and riboseq, but we also do using our approach to crack the Massback data.
Okay, got it. Thank you.
So by the way, Klaus, we recently generate a proteome data for BRCA6 and D2 after a salmonella infection. I think maybe I can share the data with you. But this is a data we generated in the UND, but it's not very deep, only about 4,000 protein. But I just kind of thought that you might be interested in this data set as well.
Yeah, we have some, so BRCA6 DBA2 comparisons, we have quite a bit for the lung. And I guess it's the lung that you're talking about.
We get it from the spleen.
All right. That is, yeah, okay. Well, I've never done, unfortunately, I probably don't have the samples anymore. We always do the lung for, okay. What kind of infection? I have to ask first, maybe.
It's a salmonella infection.
Ah, yeah, okay, okay. So I have flu and lung data. And we do have transcriptomes of the lung and of the blood. Yeah, okay, yeah, different data.
Yeah, our next experiment will do infection using salmonella, do the infection for AD mouse model and the BRCA6 mouse model, BRCA6 mice, and then we will do the proteome profiling of the infection as well.
Okay, sounds interesting. Excellent.
Thank you.
I had a quick question if there's time. I was just curious about, I think you detected between like 8,000, 9,000 proteins across your batches, and you were using the whole brain, I think. So would that number change if you looked at like a region or cell type, or what's determining that, like that number of proteins detected?
The number largely depend on how deep your fractionation. It's not depend on the brain region. Because anyway, any brain region has more than 10,000 genes, right, or proteins. So if we go to deeper enough profiling, and we should be able to detect the large amount of protein. And I would say it's really depend on how deeper you are going to do.
Do you have a sense for sort of the upper limit right now of protein detection?
Podium, we would say for our Genomics paper, this is the deepest podium we ever get it. And this is 14,000 proteins from cortex.
Thank you, that's awesome.
And for the phosphorylation, we get the deepest data is the 100,000 phosphorylation site.
One of the things I was struck by, Xusheng, was the number of trans eqtls compared to the number of cis eqtls that you found in the rat. Did you do a similar protein-QTL study in the human data that you had?
Yeah, we do. We do.
Did you see that same ratio of trans to cis?
Well, we get, for the human, we get around 500 cis, and we get about 200 trans. We didn't do too much analysis about trans. We still doubt about the trans. So, yeah.
Any further questions from the audience?
I had a question for Rob, but he's gone. He said that data is not sacrosanct. Please feel free to fiddle with it. I think that, at least on the face of it, that sounds extremely dangerous. But you know, maybe perhaps he meant something else.
All right. Well, if there's no further questions, then we'll thank Xusheng for a great presentation and thank Rob in absentee for his presentation. The video is being recorded. We will have that up on YouTube as soon as possible, along with slides. And I saved that document already that Rob shared, and hopefully with his permission and with Xusheng's permission, we'll share that also on the OPAR website, so you can have that walk through along with the presentation.
All right. Thank you for having me. Thank you.
Thank you.